Now look at this.
What's up, Epipella Penguins? Back again for 5.3 part 2 because Miss Jones talks too much. So there's different types of dominance, and that's one thing we need to focus on. So the first kind we have is complete dominance. That's what we saw in the previous video, in which if you have at least one dominant allele, it's going to completely mask the recessive allele. Wait, what did you just talk about, Miss Jones? That makes no sense. So dominance is based on the fact that the dominant allele is going to produce a functional enzyme, functional protein. It's going to make something. Whereas the recessive allele traditionally is going to either not make something or it makes a non-functional enzyme or non-functional protein. And so if it's a complete dominance, we see that one allele is going to be able to mask or cover up that recessive allele. We also see that the complete dominance, that the single allele can make enough of the product that you're going to get the same phenotype between homozygous dominant and heterozygous. Incomplete dominance traditionally has to do with blending. So neither one is completely dominant over the other. So if I have, red snapdragons and white snapdragons, the hybrid of those, or the heterozygous of those, is going to be a pink snapdragon because it makes the red pigment, the white pigment, and those mixed together giving us the pink pigment. Codominance has to do with, again, neither one is more dominant than the other. But you're going to see each dominant trait as its own distinct entity. So AB blood is a perfect example of this. So if you have the A allele, you make the A glycolipid. If you have the B allele, you make the B glycolipid. So if you have AB blood, you have A allele and the B allele, then you have AB blood because you have both the A glycolipid and the B glycolipid at the same time. And so because incomplete dominance and codominance are going to have a different phenotype with the heterozygous, in these cases with a monohybrid cross, I'll see a 1 to 2 to 1 ratio instead of the traditional 3 to 1 ratio. Sometimes the genetic problems are going to require us to work backwards. So in that case, if I have a 1 to 1 ratio, that tells me that one parent must be heterozygous and the other parent is homozygous recessive. That's the only way I can get a 1 to 1 ratio. I think it's important to note that just because something's dominant doesn't mean that it's prevalent. So polydactyly is an example of a dominant trait. I have 5 fingers, so I'm going to be homozygous recessive. But an individual that has the dominant allele for polydactyly is traditionally going to have more than 5 fingers. They might have 6. So here's a clip of a family that does have polydactyly.
Meet the De Silva family.
Hope that was helpful. Don't forget to hit that follow button. And remember, 8th Bad Pain, success. Bye, y'all.